Hey guys, welcome back to biology. Today's lesson is going to look a little bit different than usual because we're focusing on using a skill rather than learning new content. So to get our lesson started, you guys should actually go take this quizzes. It's going to be assigned in a separate post on Classroom. It's scheduled to post at 7:30 a.m., but it ends on Thursday. So if you don't take it before Thursday, you won't have the option to take the quiz. Make sure you take it today or tomorrow morning, depending on when you get around to the assignment, but it has to be done before Thursday at noon. So make sure you do that. Go ahead and pause the video, go take that quizzes, and then come back and we'll get started with today's work. Okay, so to get us started with today's work, you guys will have this attached as a PDF. I want you to go ahead and open this because this is the crux of our lesson for today. This is what we are focusing on. So you guys are going to learn how to read codon charts like this and a codon wheel, which looks like this. The reason this is an important skill is we've been talking about the central dogma and how protein is synthesized. And remember, during translation, we go from an mRNA codon, which is 3 letters, to an amino acid. And this tells us how to predict what amino acid is going to be produced based on the codons provided. So you guys should split screen between this and my video. I am going to use the one that's on your independent practice just because I don't have access to these drawing tools if I'm on a PDF. So you should keep this one pulled up and then watch the video. I'm going to walk you through how to do the practice. And then the only work you need to turn in is the practice, your exit ticket, and your quizzes. So make sure you turn all 3 of those in for today. So let's get started. Today we're going to talk about how to interpret codons into the amino acids they produce. Now to do that, biologists use one of 2 things. They can use this codon chart right here or the codon wheel. I'm going to teach you how to read both, and then you can decide which one is easier for you and use that for the remainder of the lesson. On your exit ticket, I ask you to use each one. For 1 and 2, you would use the wheel. For 3 and 4, you would use the chart. If you're uncomfortable with that, use whichever one you understand the easiest and use those the whole time. So when we're talking about codons, remember those are groups of 3 letters or bases on nucleotides. And we are going to take those 3 letters and then determine which amino acid is produced from them. All right, so for our practice, we are going to focus on this first one right here. If we had the codon CCC, we want to know which amino acid it would produce. Now, if we're using this one on the left, it's not labeled on your chart right here, but it is labeled on the practice, the order in which you would use it. So our first base in the codon, that's the first letter, is on the left-hand side here. Now, our first base, we know, is a C. So we're going to go to this position. Now we're going to triangulate using the second base across the top. So our second base is also C. Now what we do is we look at where the first base and second base intersect on here. So if we were following it down to here, follow it over, see where they overlap. So it's going to be one of these 4 right here. Now, for your third base, it's based on which quadrant you end up in. So we know that we are in this box right here. We're going to go look, C is right here, so we'll follow that back over. And we know that for number 1, the first one, it's going to produce PRO. So in the box right here, you would just type PRO. I'm going to leave this in and we'll do one more together and then you guys can practice on your own. So for the second one, I'm going to use the wheel over here. And we'll use this one right here. So we're going to focus on A, G, and U. So to use the wheel, it's much simpler. We just start in the center and then work our way out based on the letters that we have. So I'm going to start with A because that's my first base in the codon. Then I'm going to move to G from there. So I go from A over to G. And then I know my last base is a U, so I end up in this purple box, and it tells me that SER right here is the one that's produced. So we'll go ahead, type that in our box right here, so we know SER is what's produced. Now again, I'm going to walk you through how to use these, and then you can do the rest of number 1 on your own. So if you are using the codon chart, remember the first base is a row, the second base is a column, the third base is within the row and column that you specify. So if we're looking at this one here, CAG, we would go to C, and then to A, see where they overlap, which is right here. And then we know our final letter is G, so it will produce GLN. Now we can do the same thing over here. So we have a C, then we go to A, then we go to G, and we see that it produces GLN. So both of these can be used to identify each of the different codons that are produced in number 1. Now let's look at number 2. Number 2 asks us to do the opposite. So instead of starting with a codon and ending with an amino acid, it tells us the amino acid that's produced and then asks us which codons can produce it. So if we look at this first one here, proline, what you should make note of is that only the first 3 letters are going to show up on the amino acid chart. So if we go here, we've got proline, and I bolded these for you so you could see which ones we're looking at. So for proline, it's PRO. We can go back up here, and you can look here, or if you'd like, remember you can go over here, look around the wheel, and find PRO. Now I think for the purpose of going from amino acid to a codon, it's easier to use this one just because they're all grouped for you, right? So when we find PRO, we've got 4 options. We've got CCU, CCC, CCA, and CCG. Now if you want to use the wheel, you can, but in order to do that, you kind of have to work backwards. So we would have CCU, CCA, CCG, CCC, all producing PRO. Again, it's your choice which one you use, but for number 2, I suggest using the codon chart just because they're grouped by the letters for you and it's easier to see. So in this box right here, I would put all of those different combinations that give us pro. So you guys can go ahead and do the other 5. You can pause the video, and then I'm going to walk you through our last couple of ones here. All right, so moving on to number 3, it asks you to identify the start codon. The cool thing about amino acids is that every single amino acid strand that eventually produces a protein starts with this specific codon. So if you look at the chart, you want to find the word start, and then put that codon in. So there's only one start codon, but there are actually 3 stop codons. So go ahead and go back to the chart and find the 3 stop codons, and you'll list those right here. Now, for this last part in your practice, we have one added step. Before, we were looking at our RNA strand in groups of codons and figuring out the amino acids produced. Now we have to take our DNA first and turn it into RNA. And if you've forgotten how to do that, you can go back to our previous examples to get help with that. So remember when we talked about transcription, we talked about how to convert DNA to RNA. So it's the same as the rules for replication. Remember, A pairs with U, G pairs with C. The only difference is for RNA, there are no T's, right? So T is going to pair with A, while A will pair with U. And then C pairs with G, just like in DNA replication. So since we've got this codon here, we would go back up to our chart or our wheel, and I'm going to go to the chart for this one and figure out what amino acid gets produced from it. So we had A and U, which puts us in this box here. And then our final letter was G. So if we follow that back over, we get AUG, which codes for methionine, which is our start codon. I'm just going to write MET right here. Now we're going to do the same thing for this next strand. So if we look, we have a C. Remember that pairs with G. We have an A that pairs with U. And we have a T which pairs with A. So we'll use the wheel for this next one. We've got GUA, G to U to A gives us VAL. So we would just type that right here. That's all you need to do for the remainder of your independent practice. And you can go back and rewatch this video. Remember, you can pause, you can start again, you can do it at slower or faster speed, depending on how comfortable you are with this. But this is a skill that you'll want to master in order to complete your exit ticket successfully. Remember, if you have questions, you can always show up to office hours, but after you finish this independent practice, and I'm gonna leave these examples in so you can see what we did, then you're going to go and complete your exit ticket. You can use the wheel and chart in here, or you can use this one that you have a PDF of, whichever you're more comfortable with. But at the end, go ahead and turn in your practice and your exit ticket. And if you didn't, make sure you go take that protein synthesis quizzes. Good to see you guys in class again, and we'll pick this up on Thursday with some new material.